Hi, good afternoon. I'm Dr. John James Wilson. I'm the Curator of Vertebrate Zoology at World Museum, part of National Museums Liverpool. Like most people, I'm currently working from home, but picture here is where I'm normally based at World Museum in my office, and the collection is down here in the basement of the building. And as I said, I manage the use and the care of the vertebrate zoology collection, which totals around 80,000 specimens of birds, mammals, fish, reptiles, and amphibians. Our bird collection is particularly important, being the second largest in the UK and being rich in specimens that are over 150 years old, including a large number of type specimens. So obviously, I'm very interested in taxonomy and biodiversity, and the message really is that there are so many species, so little time. There are estimated to be at least 10 to 15 million animal species on our planet, of which around only 1.5 million have been scientifically described by taxonomists. After several years of training, typical taxonomists can perhaps become an expert on around 1,000 species, which means we need at least 15,000 active taxonomists with their experience evenly distributed across the animal kingdom to have an effective identification capacity for animal species. And that's talking on a global scale, assuming that everyone works together. So yeah, it's called the taxonomic impediment, and it's a real problem in terms of identification of species, is the lack of taxonomic expertise. So I'm here to talk today about DNA barcoding. And the story goes that Canadian professor Paul Hibert was walking around his local supermarket thinking about the moths in his back garden when an idea struck him. The supermarket cashier can accurately identify thousands of consumer products and their prices through the universal product codes, the barcodes that are present on each item. So Paul was thinking, can we use barcodes to identify those moths in his back garden? And in particular, can we use DNA like barcodes? So here's Paul. This is what he really looks like. And he was actually my PhD supervisor at the University of Guelph in Ontario. Paul, he wasn't the first to think about using DNA for species identification, but was the one who popularized the term DNA barcodes and DNA barcoding and brought it to the public's attention. So the idea is basically that just as species show small, consistent differences in their morphology that enable us to tell them apart, as you can see here from these grass yellow butterflies, there's small, consistent differences in the marking on the underside of the wing. And just as they differ in these ways, they also differ in their DNA sequences. So let's think about how we might use DNA as species barcodes. As you might remember from school, DNA is a double helix made up of chains of paired nucleotide bases. These are adenine, cytosine, guanine, and thymine. So now you might be thinking, how much DNA is in a cell? There's got to be a lot, right? And wouldn't it actually be making the problem more complex to use DNA for species identification? Well, yes, there is a lot of DNA in a cell. So for example, the human genome is made up of 46 chromosomes, which include around 20 to 25,000 genes in about 3.2 billion bases. So the idea of barcoding is that we can focus on just one of these genes, and that gene must have the required properties of a barcode, in that it is identical, the sequence of that gene is identical or very similar in all members of a species, but is very different, well, relatively very different between individuals of different species. So that's what we need from a DNA barcode. Based on previous findings, Paul knew the mitochondria would be a good place to search for a gene that could function as a DNA barcode. The mitochondria is an organelle within the cell. It's known as the powerhouse of the cell. And this organelle has its own small amount of DNA due to its evolutionary history. It's got a small genome of its own. There's several mitochondria within each cell. Within the mitochondrial DNA itself is also found in several copies within each mitochondria. And it's relatively simple structure, being a short circular piece of DNA with around 37 genes only. So we've already simplified the problem quite significantly from 25,000 genes down to 37 genes. Within the mitochondrial DNA itself, it was decided that the cytochrome C oxidase 1 gene, known as CO1 gene, would be the best candidate for a DNA barcode. And in fact, this is the DNA barcode region that we use for animals. The DNA barcode itself actually is just about half of the CO1 gene. That's approximately 658 base pairs only. So there's the DNA barcode location within the circular mitochondrial DNA. Yep, so in effect, DNA barcode is just 658 base pairs of mitochondrial DNA. And it can be shown simply as a string of letters with only 4 possible combinations, A, C, G, or T. And this here is a representation of the DNA barcode of the very toxic, venomous brown widow spider, which is actually spreading around the world. So how can we use these letters to identify species? How does it work? So what happens is that you compare the DNA barcode from your unknown specimen. Say you've got the leg of an unknown insect, you get the DNA barcode for this, and you compare it against a reference library of DNA barcodes from specimens with confirmed species names, and you look for a match. So you can compare the similarity between this unknown barcode, this query, with each of the reference library barcodes, get a percentage similarity, and you're looking for a match. So if the unknown barcode matches with a barcode in the reference library, you can be confident that your unknown specimen is a member of that species. So there you go. That's a 100% match to this particular sphingid moth. So now we know how it works. Question is, how do we get those letters in the first place? And as any of you who have joined the DNA barcoding workshop will know, we get there through lots and lots of pipetting. These are the basic steps on the way to producing a DNA barcode, and I'm going to go through each of these briefly in turn. So the first step is DNA extraction, which involves cell lysis or cell digestion, and then purification of the DNA. Cell lysis is breaking open the cell and its contents, as you can see from the animation here, releasing the cell, including the DNA, into a solution. Then purification involves binding of that DNA so that all the other cell components or the parts you don't need are washed away. The DNA is isolated using a filter, and then the DNA is released from that filter into a pure solution of DNA, purified DNA. So that's what this diagram shows at the bottom here, the purification process using a filter. There are commercially produced kits available for this DNA extraction step, and this is the one that we're currently using at the museum. So next we need to isolate and copy the CO1 gene fragment that is our DNA barcode. And for this we use PCR, polymerase chain reaction. PCR involves pipetting the purified DNA, primers, nucleotides, and the polymerase enzyme into a microtube, and then cycling this tube and its contents through a set of different temperatures in a PCR machine, which is also known as a thermocycler. So yeah, you pipette these into a tube and then put it into the thermocycler where it will be cycled through a range of different temperatures. So in the first step in PCR, the double helix of DNA separates into the single strands. These primers, which are specifically designed to target the DNA sequence that we're interested in, our CO1 DNA barcode, the primers will attach to that specific region of DNA, and then the nucleotide, the polymerase enzymes will join onto the strands and add nucleotides to the target sequence, effectively creating a new copy of that sequence. And then this is repeated again. So in the next cycle, more copies of the target DNA are created through the primer binding and the polymerase adding those nucleotides, copying the sequence. So this is repeated 30 to 40 times, 30 to 40 cycles. So millions of copies of the target DNA are created. Again, commercial kits are available for PCR. So these contain all of the ingredients that are needed except for the purified DNA and the specific primers. And this is the particular one that we've been using at the museum for our DNA barcoding. The next step is DNA sequencing, where our target DNA is read. Okay, so we use Sanger sequencing generally for our DNA barcoding that we're doing at the museum. In Sanger sequencing, the PCR copies are terminated at different lengths with fluorescent nucleotides. So this results in every base pair in the target sequence being tagged with its corresponding color corresponding to which particular nucleotide base, A, C, G, or T, is at that position in the sequence. This is then passed down a capillary where there's a camera that captures the fluorescence. So as the sequence passes down, the camera is capturing the fluorescence at that particular point in the sequence, and it will result in a chromatograph indicating the different nucleotide base at each position. So we'll get a chromatogram 600 to 700 base pairs long, like this, a graph, different peaks of color indicating the different nucleotide bases in each position. So we don't have a DNA sequencer at the museum, and we send our PCR products to the facility at the University of Sheffield for Sanger sequencing. The chromatograms are sent back to us by email, and then a computer program can be used to convert those peaks, the graph, into letters, basically into the nucleotides. So obviously here the black color is representing G, the green color A, the red color T, and so on. So all the letters can be assigned to each of those different peaks to get the nucleotide sequence in the form of letters, like what we need for our DNA barcoding. Always worth to check that the computer is making the correct calls for each of those different peaks. And this step is known as sequence editing. So it simply involves checking the calls and making a judgment call yourself based on the height and the cleanness of each of the chromatogram peaks, in case the computer is making any errors. But this is a very straightforward step usually. So after the sequence editing process, we'll end up with our 650 or so letters and can proceed to identify the sequence and our unknown specimen. So as I explained earlier, this can be done through making comparisons with the reference library. And in reality, this is an automated process. So we'll just simply paste 650 letters into the Barcode of Life data systems identification, which is available freely online. We'll just paste our sequence into there. And bold will compare our sequence with the nearly 4 million sequences that are present in the library and then suggest an identification. So for example, this sequence here has a 100% match to several sequences of this species of geometric moth. Right, so some of you might have been paying really close attention, and you'll have noticed that bold currently has about 4 million sequences. But I told you earlier, there are around 10 to 15 million species of animals on Earth. So obviously not every species is yet included in bold. And it may be that you don't get a close match to anything in the library. It could be a result like this. In this case, bold will give the sequence a barcode index number, known as a bin. And these numbers can function as the same way as traditional species names, in that it's stable, persistent, and interoperable as an identifier. And you can use this until a name can be assigned to that barcode in that specimen. So that might be when the specimen is morphologically identified. It could be described as new if it's a new species, or it could, as new sequences are added to bold, it could be matched with named sequences later. Then your sequence will eventually get a taxonomic name. Okay, so that covers the laboratory and online steps required to do DNA barcoding. These are the standard methods, and as you can imagine, they're constantly being refined, evolved, and developed to become more practical. And one major push has been moving this work out of the library and into the field, so where exactly it is needed. So you can do pipetting, DNA barcoding in the field or in the forest, as this picture here shows. Companies are developing portable laboratory equipment to take with you into the field, such as this bento lab, which is about a laptop-sized mini lab, which contains a small PCR machine, centrifuge, and all the other things you need for DNA barcoding in the field. There's even portable DNA sequencers being developed. So it's important to note these are using a very different technology compared to the Sanger sequencing that I've explained earlier. So some very significant developments going on in moving DNA barcoding from the lab directly into the field. So that's all well and good, but you might be wondering about DNA barcoding in practice. So I'm very briefly going to introduce a few projects, DNA barcoding projects that I've been involved with in Malaysia. So there's been a lot of interest recently in using environmental DNA for species monitoring. This is where trace DNA present in the environment can be used to detect a species that's present at that environment without a need to capture or even see it. Okay, so we've done a pilot study using environmental DNA to detect this species. Any ideas what this is? Well, it's actually a southern river terrapin. Okay, so this southern river terrapin, as it's swimming along, like all organisms, it will be shedding DNA into its environment through feces, pieces of skin, and other secretions. So there's DNA present from this organism in its environment, which in this case is the river. And if we want to know if that species is present, we can take a sample of the river water, extract DNA from the water, do PCR, that's specifically for our terrapin DNA barcode, and sequence that terrapin's DNA barcode to determine its presence in the river. These kind of methods are especially useful for species like the southern river terrapin, which is very rare and elusive, very hard to see, visually monitor in the river. So we tested this method using a short DNA barcode, actually 110 base pairs of mitochondrial DNA. We tested it in vitro, taking water from the turtle hatchery where certainly turtles present. And we also tested it in situ in the Common River. And we managed to detect terrapin DNA from this river water. So another similar but slightly different idea is using invertebrate-derived DNA, iDNA, it's also known as. And using this approach, we can detect mammal DNA from the guts of blowflies. So mammals also are very hard to detect visually, to find in the rainforest. And the blowflies, which of course are relying on mammals for their nutrition, they're feeding on mammal carcasses, wounds, or feces. They're much better at finding mammals in the forest than we are. So we discovered that mammal DNA will persist in blowfly guts for about 72 hours. The flies travel about 1 kilometer a day, which can help us pinpoint the locality of those mammal species. And we designed PCR target that will preferentially copy the mammal DNA rather than the flies' DNA, which of course aids with the DNA sequencing. We tested our fly trapping and sequencing protocol against the common methods of surveying mammals in forests. So this includes camera traps, mist nets and harp traps for bats, cage traps and hair traps. And we found more mammal species with the flies than with these other methods. So it's quite a simple procedure. You can set these baited traps with some rotting mammal flesh in here to attract the flies. They fly in, get trapped in this area, and then we can take the flies, extract the gut contents, sequence those gut contents, and detect mammal DNA. So then we can determine which mammals are present within that forest area. Another area of advanced DNA barcoding is known as DNA metabarcoding. This is where large mixed species samples, such as those collected by malaise traps, are sequenced simultaneously using high throughput sequencing. And we use this method in Peninsular Malaysia to compare seasonal species turnover in different forest types, such as oil palm plantation as shown here, which of course are expanding in Southeast Asia and replacing more natural forest types. For DNA metabarcoding, it means that this large collection of insects, such as you get from a malaise trap, don't have to be individually sorted. The DNA can be extracted in bulk. You can do the PCR in bulk, just based on that bulk mixed species DNA extraction. And then you can sequence this PCR product using next generation technology, such as the Illumina MiSeq, which should theoretically produce sequences, barcodes, for all the species in the samples, which can then be sorted bioinformatically rather than physically. We also used a similar approach to sequence another type of complex samples, bat guano. So this is a famous site in Malaysia called the Batu Caves, and there's a colony of bats roosting in this cave here. So this bat guano from these bats, it contains DNA from many species of plants. We did a plant barcoding project to see which species of plants these bats are feeding on, and also which they're potentially pollinating. It's interesting then, because if you know the distribution of the plants, you know how far the bats are traveling in order to feed out of their roosting colonies in this cave. It's another interesting project, but let's move a little closer to home and see how you could use DNA barcoding in your recording activities. So I think it is fair to say that the UK is slightly behind other equivalent developed nations in terms of building up its DNA barcode library. And local recorders and enthusiasts can be the drivers of DNA barcode library building here in the UK. Together with the Tanyptera project, World Museum has started running DNA barcoding workshops where you could use DNA barcodes to confirm the presence of new or rare species within your area. So a good example was this blood bee, a new record for Cheshire identified by Chloe at our first DNA barcoding workshop. And I'm sure there are many examples where barcoding could be very helpful to you to confirm species records. So the NBN Atlas is a record of species occurrences in the UK, which I'm sure you're all contributing to through iRecord. And the Darwin Tree of Life project, which is a very ambitious project to sequence all the species of multicellular life found in the UK, they're using this NBN Atlas to direct their sequencing of certain taxa, to direct which taxa they should be sequencing. As part of this project to build up the library of DNA sequences for all the species in the UK. So if you're contributing to iRecord, you're also indirectly contributing to this project, which is known as the Darwin Tree of Life project. It's run by the Wellcome Trust and the Natural History Museum in London. Okay, so now you know how you can get involved, and hopefully barcoding is no longer an alien concept. It's clear how simple actually and how effective barcoding can be. And I hope now that you're full of ideas of what kinds of specimens you'd like to DNA barcode yourself. Happy to take any questions. And also, just before I finish, I'd like to acknowledge, of course, there's lots of other people who have been involved in the projects that I've briefly covered in this talk, including, of course, my co-authors in my publications, which you can find through my ResearchGate profile, and where, of course, the funding sources for those projects are also acknowledged. So yeah, I'm happy to take any questions. And of course, if you prefer, you can always get in touch with me through Twitter or through email. Okay. Thank you very much.
Thank you, John. That was really interesting. So if anyone does have a question, you can either put your hand up virtually or you can ask a question in the chat. We've got one question from Mike Ashworth. Does the material have to be fresh, or will barcoding work on insect specimens which have been dried for days, months, or years?
Yeah, good question. So obviously fresh material is better, but it can work with material that's been dried for 100 years. You can get DNA barcodes from that kind of material as well. Depends on how they've been preserved. Yeah, but it's possible.
Okay. Does anybody else have a question? Everyone seems quite quiet today. Maybe it's because we're in the evening today. Glenn, you got your hand up. Do you want to unmute yourself and ask a question?
Yeah, just a quick question, John. What are the problems of cross-contamination of some of these DNA sequences?
So,
how do you prevent it occurring?
Yeah, it can occur. The idea would be that if you're taking, say, for example, a leg from an insect in order to sequence that, maybe you've touched another insect previously and then you've touched that leg. Is that the kind of contamination you're thinking about? Yeah. So there should be much more of the DNA of the specimen that you want to sequence in that DNA extract. There may be a small amount of DNA from another species, but the PCR will pick up the DNA that's most abundant. So if your target has more DNA there, that's what will get PCR.
So it's just the biggest percentage of the DNA.
Yeah. It could be that if the contaminant might show up as those chromatograms that I showed you, you might see a faint bit of background noise that could represent the species that's caused the contamination. Yeah. So it's better to try to avoid it in the lab in terms of taking, taking those kind of precautions to prevent contamination.
Unless the cross-contamination is a species that's not been recorded before.
Yeah.
Okay, thanks very much, John.
Okay, thank you. We've got another question in the chat. What particular primers do you use?
Yeah, so there's good primers that have been developed for insects. So they've been around since I think about the 90s actually. Yeah, and they've been found to be very effective across most animals in a way, but there may be some species that they don't work for, so you might need to modify those primers slightly to be able to target different groups of species.
Okay. Got a question from Diana. Is it possible to get estimates of abundance from the Metabarcoding studies?
A good question. There's been a lot of work looking into that because of course it would be very useful to have that kind of information on abundances. I think lately some of the papers that I've been reading, they have shown that they can estimate abundances from metabarcoding data, but it requires kind of a more complicated experimental design in order to achieve that. And things like putting in known amounts of different species to be able to calibrate it, things like that need to be done in order to get reliable abundance information from metabarcoding data. Yeah.
Okay, thank you. Yeah, we have another question from Vieteki. Sorry if I'm pronouncing your name wrong there, but she says, in terms of storage of specimens samples to be used for barcoding, have you found any differences in using specimens in ethanol versus other preservatives in terms of the success of amplification?
Yeah, so for insects, it seems to be that dried specimens actually are the best source of DNA. For other types of animals that I've been working on, ethanol is better. At the museum, we have a lot of specimens stored in IMS, so denatured ethanol. It does seem to have negative effects in terms of the DNA quality. So maybe the DNA is degraded, not so easy to amplify with PCR. But that's just anecdotal. I think there are some studies that have looked into that, which is the best way to preserve them. But a lot of my projects I've been working with very fresh materials, such as village trap samples that are, just weeks old. So there haven't been any issues with that. But yeah, now I'm looking more into the historical material at the museum. It does seem to be quite an issue with the way that they've been stored, different types of fluids have been stored in.
Yeah. We have a question from Jeff Garnus. He says, do you primarily / exclusively use COI for metabarcoding of insects? What do you do about COI pseudogenes in the nuclear genome? And is there anything that can be done for groups where COI doesn't resolve? Quite a few questions in one there.
Right. So some people have been using 16S, which is another mitochondrial gene for metabarcoding. The disadvantage with that is that several species may share the same 16S sequence, whereas CO1 tends to be that each species has a distinctive sequence, so it actually functions better as a barcode. But 16S has other advantages that maybe it's easier to amplify, kind of thing. So generally, I would suggest using CO1 as the barcode. There was a few other questions in there as well. Oh, the pseudogenes. Yeah, it's more difficult with metabarcoding to detect those pseudogenes. So there's, as you might know, there's different ways of detecting them based on translating into protein. If there's errors in terms of the translation, that suggests that the sequence could be a pseudogene. But yeah, and there was a third part as well, wasn't there?
Yeah, is there anything that can be done for groups where CO1 doesn't resolve?
Doesn't resolve. Yeah, looking into other genes, really. It's interesting. It would be an interesting case if CO1 doesn't separate the species. What is the reason for that? Are they relatively young species? Those kind of questions then come up, which are interesting in themselves as well. Yeah, if you have that situation.
Okay. From Rod Hill, why use only one region of the gene, i.e. can you use a second short region to validate the screening, or is this specificity of the selected region sufficient to offer the precision and selectivity required?
Yeah, so it's a good question as well. As I kind of pointed out in my talk, the DNA barcoding, the whole idea is to try and make it as simple as possible. So that's the reason to focus on a single gene region. Why make it more complicated if this single gene region can work? And it tends to be that CO1 is very effective as a DNA barcoding for most species of animals. Of course, if you have further resources, more funding, or there's some reason why you're suspicious of the result of the CO1 barcoding, you could always look at other genes as well. So there are other genes that also could work well as a DNA barcode. Primers might not be as well developed, particularly in terms of nuclear genes. There's other issues. In terms of mitochondrial DNA is present in much higher abundances within a cell because it's several mitochondria and there's several copies of the mitochondrial genome within each cell, whereas there's only a single copy of the nuclear DNA. So it has advantages in terms of that as well. But yeah, definitely you could look at other gene regions. Then if you've got different results from different genes, that also raises some interesting questions as well. Is it a hybrid, that kind of thing?
Thanks, John. Katie Woodcock is asking, have you stored malaise catches for periods of time to extract at a later date? And if so, what did you find to be the best in terms of preservatives and temperature? And also, can I ask what preservative you used in your malaise trap catch bottle? What you found to be the best? Thank you.
Okay, so we used ethanol in the bottle for the catch, and also that's how we preserved it. Then we put it into the freezer at -20, but we didn't store them for that long. I'd say less than a year in all cases. So in terms of longer periods of storage than that, I think probably the -20 in the freezer in ethanol would be suitable. I mean, if you do have access to a freezer that goes to lower temperatures, then the lower the better, really. But that I think would last for several years at that kind of temperature and in ethanol.
Okay. Mark Sheridan is asking, as a moth trapper, could this provide an alternative to Gendebt for difficult species by using just the scales?
I'm not sure I totally understand the question. So it's
so an alternative to like using genital determination, I think.
Yeah, okay, yeah. I think so, yes. Definitely. Yeah.
Okay, we've got a question from Chris Workman. If the barcodes are based on the single mitochondrial gene cytochrome oxidase with 650 nucleotide / possible variation, how much overlap or how many variations can show up between the species?
Right, so it's generally about 2%. Different is used as a very crude threshold. So if it's within, if your sequence of your unknown is within about 2% of one of those reference library sequences, you could assume that it's the same species. If it's over 2%, it's suggesting that it's a different species. So that's what's used as a crude cutoff. But what we actually see generally is that there's a barcoding gap. So there's usually the variation is within 1%, and then it's probably above 5% to the nearest neighbor species, so to the next species, you're going to be looking at those kind of values for similarity.
Thank you. A question from Laura Weldon. What software do you use to process and sort your data? Can you recommend / do you have a preference for metabarcoding data?
Metabarcoding data, even for metabarcoding data, I find the Barcode of Life data systems to be the best place for storing data. It's online, so you can access it from anywhere. You don't need to have a large amount of storage on your own personal computer. So that's what I would use, even for metabarcoding data. Obviously, you have if you're going to publish the data, ultimately you'll need to submit your reads to Gendebt, that kind of thing. But yeah, both can be useful for metabarcoding data as well.
Yeah, she says thank you. Any more questions? If anyone's got their hand up. No. So yeah, if nobody has any more questions, yes, I'd just like to say thank you again to John. And thank you everyone for joining us today. And this has been our first evening webinar, and it's gone quite well. So if you would like to watch the presentation again, we'll be uploading this to YouTube within the next week, and I'll send out a link to that and some various links as well. So our next webinar is this Saturday, which is on water beetles. So please do join us for that if you can. That one will be at lunchtime at 1 o'clock. So yeah, thank you and good night, everybody.